Okay, so this is essentially part 2 of transcription. Eukaryotic gene transcription. Above we talked about is prokaryotic gene transcription. Now we talk about eukaryotic gene transcription. Essentially all the steps are the same, but there are certain factors that are more there are more factors, and because in eukaryotes there is more regulation, and that occurs with different types of proteins involved. So we look at that. So in chromatin remodeling, so basically what happens, that this is your cell, right? This is your nucleus, and then everything is, the chromatin is here. It is highly compact. You cannot, in order for transcription, you need a long strand of DNA molecule, right? So what actually happens is that we have a good equilibrium between euchromatin and heterochromatin, right? Euchromatin is loose DNA, and that is compact. So what is chromatin remodeling is that the reversible nature of euchromatin and heterochromatin. So we need loose DNA for gene transcription. How do we do that? How do we do that? Euchromatin via methylation of DNA, right? What happens is that DNA is methylated, more CH3. More CH3 means lack of positive charge, and then we cannot attract it to histone, and it will become free. That's it. That's the main crux of it. Then we have basically 3 types of RNA polymerases: RNA pol 1 for rRNA, 2 for mRNA, and 3 for tRNA. But in prokaryotic, we only have RNA polymerase. There are no numbers. One RNA polymerase does everything, but in eukaryotes, there are 3 different types. Now, the promoter regions for pol 2 are Tata box, and we call it as the Hogness box. The Pribno box is present in the prokaryotic transcription, and Hogness box is in eukaryotes. And there is also initiator regions, or it could be downstream promoter elements. Downstream promoter elements is located in the positive integers, after the start of the transcription. That's why they're called downstream, right? But most important is the Tata box, that is -25 upstream. Okay, that's important to remember. And it is cis-acting. Cis-acting means because they are on the same molecule, same DNA molecule they are located, that's why they are cis-acting. Okay, now important thing is general transcription factors. The general transcription factors are proteins. They are, of course, encoded by different genes, and they are synthesized in the cytosol. And then they are again brought back into the nucleus. That is why we call them transacting. They are not on the same molecule, they are from a different site, and they come for you in the nucleus. There are different types. Let's look at some examples. These are important examples. We have TF2D. So this is 2 basically, TF2D. TF2D recognizes the Tata box. TF2F brings polymerase to the promoter, and TF2H has the helicase activity to melt DNA, and kinase activity to activate the polymerase. This activation of polymerase will let the RNA polymerase to clear the promoter region and slide along the DNA for transcription, right? So these are points to remember, and some mnemonics you could do is the H is for helicase, and D is for D, and T has the same kind of sound, so D T D T you could say like that. and the last one is polymerase. So TF2F it brings polymerase to the promoter. Then there are certain regulatory elements. The regulatory elements, it could be in they could be proximal or they could be distal. They could be near, like for example, what does proximal and distal means? This is the DNA, like this, right? Not like that, wait, like this. So this is our a region of transcription. So things that is near to the transcription region is called as proximal, and this bending region right here, this is, see, this is how far this is, how far this is. This region is distal. This is going to activate this or enhance this or activate this or mediate it or inhibit it, right? Anything that how regulation is done. So mostly distal regulatory elements, they are enhancers, and proximal, the examples are CAT and GC boxes. Now here is the special transcription factors. The special transcription factors have two domains. One is the DNA binding domain and the other is the transcription activation domain. So if this is the DNA and this is the special transcription factor, it will have a DNA binding domain. So this is the DNA binding domain, and it is going to have a transcription activation domain that is going to be above it, right? And this is the STF. So transcription activation domains recruits GTF, the TF2H, TF2F and TF2Ds. These are the GTFs, the major ones, TF2F, TF2D and TF2H, right? So these are twos. That is going to help special transcription are going to help recruit them to the promoter, and also co-activates HAT enzymes. So these HAT enzymes are going to methylate DNA and forms more euchromatin, right? Pretty simple, right? Okay. And that is it. And these STFs, they are going to bind where? Into regulatory elements, okay, either at proximal region or distal region, right? Okay, one more thing here, why does STFs bring HAT enzymes? I like to a little bit elaborate on it here. You see, different kinds of cells, in different kinds of cells there are different kind of activated, like for example, this is a muscle cell, right? If this is a muscle cell, the only genes for microfilaments will be activated, right? Like actin or myosin, right? It will not activate genes for the production of any kind of enzyme, like or a hormone or an enzyme like a protein hormone like insulin. It will not have genes for that. It will have genes for that, I'm sorry, but they will be off, right? They will be present in the form of heterochromatin, not euchromatin, right? So that is the whole scenario of euchromatin, heterochromatin. So every cell has its different kind of genetic activation, right? That's why there are special transcription factors. In need, there could be some kind of cells that would not normally be producing certain types of molecules, but special transcription factors are going to go into the nucleus and they're going to activate that. Using HAT enzymes, they're going to convert heterochromatin to euchromatin, so that can be transcribed, right? If you understand this concept, it is important. Okay, that's it about eukaryotic gene transcription. Thank you.